Hello everyone. In this video, I will talk about the timescales as system-level diagnostics of the carbon cycle. In a previous video, we saw about compartmental dynamical systems as very general forms of global carbon cycle models or ecosystem carbon cycle models, in which the change over time of the mass of carbon in the compartments will be equals to the inputs of the compartments and all of the rates and transfers of carbon among different compartments. These systems can depend on time and can also depend on the mass that is stored of the different compartments. This is a very useful representation of carbon cycle models because we can easily estimate these concepts of age and transit time that I will introduce now. But before that, let's think about the heterogeneity and the complexity of biogeochemical systems. So for example, carbon and nitrogen cycle models. For the carbon cycle, carbon will enter an ecosystem through photosynthesis. It will get allocated to different plant parts. It will go to the soils. It will get to respire. And there will be a lot of complexity in an ecosystem that somehow has to be integrated in an ecosystem model. Models sometimes are very simple and sometimes they're very complex. They introduce many different processes or in some cases, very simple processes. The same for the nitrogen cycle. The nitrogen will come into an ecosystem either through biological nitrogen fixation or deposition from the atmosphere. It will enter complex pathways through food chains, and eventually that nitrogen is going to get denitrified or is going to be leached out of the system. In general, for the ecosystems, we're interested in knowing about an integrative measure that can tell us about all the different processes that are occurring simultaneously. And concepts that have to do with time are actually very useful. So let's think, for example, about the time that it will take carbon atoms to travel through the whole ecosystem since the time they are fixed by photosynthesis until they are respired back to the atmosphere. Or the same for the nitrogen cycle, how long would it take for nitrogen that is fixed to travel through the whole ecosystem until it is lost. And for that, we use these concepts of ages and transit times. To understand these concepts, let's think about them in a very abstract, general way. Let's think about a system, such as an ecosystem, that has multiple compartments. And this system stores mass, but we're going to think about the mass as small particles or atoms, and each of them is going to have a clock that is going to measure the time that the particle stays in inside the system since the time it enters until some time of observation. So all the mass that enters the ecosystem will have an age equals to 0. When they enter the system, they start mixing with other particles that have been there already in the system that have different ages, and then therefore all of these particles are going to have a mix of ages. Now, we define system age as the age of all the particles inside the system since the time they enter until some point of observation when we look at all of these particles in the system. If we collect all of these particles, we can, for example, do frequency distribution with all of them to look at the age frequency distribution, and we can compute the mean age of all these particles. We can compute the standard deviation of the ages. And therefore, we obtain a good statistical characterization of the ages of the system age of carbon inside our ecosystem. We can do the same for the concept of pool age. If instead of looking at all the ecosystem, but we focus only on one single pool, and we collect all of the particles inside the pool, we look at the age, and we look at a frequency distribution, at a mean, at a standard deviation, we can characterize the pool age of carbon inside the pool. Now, if we, instead of looking at the mass that is stored inside the system, but we instead look at the output flux, what is leaving the ecosystem, let's say in the respired CO2 out of the ecosystem, we can characterize what we call the transit time, which will be the age of all the particles in the output flux at the time they are leaving. The transit time is actually a good measure of how long does it take for carbon or mass to pass through the whole ecosystem since the time it enters until it leaves in the output flux. It turns out that for complex heterogeneous systems, the age and the transit time are actually very different. But for very simple systems that only have one single aggregated pool, the system age and the transit time are actually the same. But since we are dealing here with ecosystems that have multiple pools and multiple transfers and connections among them, then we need to assume that the system age and the transit time are different. And the other important thing is that we should look at entire distributions of ages or transit times. In the past, we focus a lot on mean values, but I think it's very important that we think about these concepts in terms of distributions. So for example, let's think about soil organic matter, the amount of soil organic matter in the soil. This soil organic matter will have an age, and the age of this SOM may have different distributions, but for example, the same mean. Here in this example, I have two distributions. One in blue would be a normal distribution with a mean age of 200 years versus an exponential distribution with the same mean age of 200 years. But we see, for example, that for the exponential case, the ages of the soil organic matter can get much larger. It could be up to 600 years if we look at the 95% quantile of this distribution. Versus for the normal distribution, the 95% quantile will be 282 years. So therefore, if the soil will have this exponential distribution, it can have carbon that is much older than if you will have this normal distribution. Also, when we look at distributions that are different than the normal distribution, we also need to think about the 50% quantile or the median of the distribution, because in many cases it can actually be very different than the mean. And the 50% quantile actually characterizes really well the central tendency of the distribution. Now, in our group, we recently developed formulas to be able to compute ages and transit times distributions for systems in equilibrium and outside equilibrium. Here in this slide, I have the formulas that we developed for systems in equilibrium. If we have an autonomous linear compartmental system that we can express in this form, we can find a steady state solution that is going to look like this, and all the output rates out of the system can be obtained as the sum of the columns of our compartmental matrix. We can obtain the system age distribution using this formula. I'm not going to go into the details of the formula, but we have all of the elements already to be able to compute that. And the mean, we can obtain it with this second formula. We can also obtain the distributions for the pools if we define a new matrix in the diagonal having all the steady state values. And we use this formula, we obtain pool ages distribution, and also we have here a formula for the means. And we can also use these formulas to obtain transit time distributions. Since systems, nonlinear systems, autonomous nonlinear systems, when they reach the equilibrium point, the mass of the system doesn't change over time, we can also use the same set of formulas for nonlinear systems that already reach an equilibrium. So this is also a very important use of these formulas, because we can use them for linear and nonlinear systems at equilibrium. For systems that are not at equilibrium and for non-autonomous systems, we have a different strategy. We can always compute age distributions as mass in the system at some time with certain age, using this double approach. On the one hand, we have to look at an initial age distribution. That would be the initial distribution of ages at time 0. that we're going to explore and we're going to move over time using this concept of the state transition operator. This is a mathematical object that is very useful, that allows us to take an initial age distribution and move it forward over time. The same state transition operator concept can be used to move all the inputs of carbon that comes into the ecosystem after our initial time zero, and look at how those inputs will change over time. And if we combine the two of them, we can obtain age distributions that move over time, that are always changing over time. So we can see here then the key concept to be able to compute these time-dependent age distributions is the concept of the state transition operator. So we need to obtain that transition operator. And for this, we develop a linearization strategy that consists of taking any nonlinear, non-autonomous dynamical compartmental systems. If we solve this system and obtain a solution, and it can be a numerical solution, we can take that solution and plug it inside the compartmental matrix, and inside the vector of inputs, we define a new vector of inputs and a new compartmental matrix, and define a new system. This system obviously will have the same solution, and because these dynamical systems only have one single solution, then both solutions will be identical. Now with this linear compartmental system, it's much easier to compute the state transition operator. The details on how to compute it are explained in this paper published in PNAS, and I encourage you to look into the details in that paper. For now, I only will show you two examples on how to use this framework. One is to look at the concept of persistence of soil organic matter in soils, and the other is to obtain the time to remove fossil fuel derived carbon from the atmosphere. So the first example will focus on autonomous dynamical systems in equilibrium, and the second example we will take a nonlinear, non-autonomous dynamical system. In soils, there has been a lot of discussion about the concept of persistence. Some years ago, there was a very influential paper about the concept of persistence of soil organic matter in ecosystems as an ecosystem property. And the main discussion in this paper had to do with the fact that if we look at very different chemical compound classes, we see that the mean residence time of carbon in these different compound classes can be very different, and things that first we thought that decompose very slowly, such as lignin, actually can have mean residence times that are shorter, and other things that, such as saccharides, can actually have longer residence times. And the idea was then, well, this mean residence time can have a lot of variability, but it's something that emerges as the effect of many different processes that occur in soil. However, one issue with these ideas is that the concept of mean residence time is sort of ambiguous, because on the one hand, we don't know if this data represents the age of carbon in soils, or if it represents the transit time of carbon in soils. And also, as I showed you before, the mean values can be quite misleading, because you can have distributions of soil organic matter that, if they have really long tails, the time that carbon can stay in the soil can actually be very, very long. So we propose that instead of looking at mean residence times as this ambiguous concept, we actually look at entire distributions, and in particular, the 95% quantile of the distributions of age, because it will tell us more about for how long or how old carbon can really get in the soil. And since we're dealing with two concepts, ages and transit times, there are three different potential relationships between these two concepts. One is if the mean age and the mean transit times, or all the age and the transit times, are similar, then we are here in this one-to-one line of the relationship between ages and transit times. And that means that the carbon that is respired from the soil has the same age as the carbon that is stored in the soil. And if we have this case, that means that the carbon that is respired has the same probability of being decomposed. And in this case, we are talking about a homogeneous system in which all the carbon stored has the same probability of being decomposed, and therefore the same probability of being released to the atmosphere. The second case is when the ages are shorter than the transit times. And in that case, the carbon will have to be retained in the system before it actually gets released. So carbon will come in, will have to stay there for some time before it's released, and therefore the transit time will be much older than the age. And the third case is when the transit time is shorter than the age. And this happens when carbon will come into the soil, it will get quickly respired, but very small amount of carbons will remain and they will accumulate over time. And therefore the total age of soil organic matter will get very old. So we were interested in a study what type of relationships we see in soils. So we use a number of models, very common models from the literature, and we use our formulas to compute the age and the transit time of carbon for these models at equilibrium. We see that the shape of these distributions are relatively similar. In case for the age distribution, we see that most of the carbon for all models is always very young, and only small proportions of carbon can get really old. And the same for the transit time. The age of the carbon that is in the release flux is always very young, but some of the carbon in the release flux can actually be very old. We see here in this graph that the transit time is in the order of some decades, and the ages can actually be in the order of centuries or maybe millennia if we actually look at the 95% quantiles of these distributions. When we do that, when we look at quantiles of the distributions and we plot the age quantiles versus the transit time quantiles, we see that we are always below the 1-to-1 line. Here, each of these lines is a different model. On the left, we have the 5% quantiles, and here to the right, we have the 95% quantiles. In all cases, we are always below the 1-to-1 line. That means that the transit times are always younger than the ages. That means that we are always in the third case, the case in which carbon comes into the soil, gets quickly respired, but some small proportions of carbon remains in the soil for very long times. And through long time accumulation, the ages of the total carbon becomes very old. And this seems to be the case for soil organic matter. This is something that we are now confirming using radiocarbon observations. And it is a very interesting system level property of the soil organic matter. We can use the same concepts to look at gradients and large scale patterns. Here, these are results from the CESM model, the version used for the CMIP5 intermodal comparison. We look at the age, or the mean age of soil organic matter across the world. And we see that in general, we're looking at mean ages in the order of a few thousand years, versus the mean transit time being only of a few decades and maybe a few centuries. In the boreal and Arctic zones, the mean transit times tend to be longer, which is something that we actually know really well. And in the rest of the world, the mean transit time seems to be just a few decades. So this is actually that has to be confirmed with more observations. We're working on that right now, but it's actually a very interesting property of the soil organic matter that can tell us a lot about many different processes that constitute the cycling of carbon at the global level. And again, if we look at the plot between mean ages versus mean transit times for models used for the CMIP5 intermodal comparison, we see that the mean ages tend to be always much, much older than the mean transit times. Now I'm going to show you a different example. This is an example looking at the global carbon cycle using a much more complex model, a model that has nonlinear interactions between the atmosphere, the terrestrial biosphere, and the surface ocean. The red lines here indicate that these fluxes can be expressed as nonlinearities between the contents of the reservoirs. This system is open with respect to the deep ocean, so carbon enters the system from the deep ocean to the surface ocean, will move to the atmosphere and to the terrestrial biosphere, and it will cycle among them. And we can also inject fossil fuels to the atmosphere to bring the system out of equilibrium. If we look the case in equilibrium before adding fossil fuels to the atmosphere, we can use our set of formulas for equilibrium to estimate the ages of carbon for these three different compartments and for the total system. Because carbon enters the surface ocean, we see that most of the carbon in the surface ocean is young. Then it goes to the atmosphere where you have a larger proportion of young carbon, but then a tail that expresses a little bit older carbon. And then the terrestrial biosphere, which is this sink compartment here, will have much older carbon. We're gonna now perturb this global carbon model by adding fossil fuels, and we're going to use an RCP 8.5 scenario just to see how we modify the age distribution of carbon in the atmosphere as we add new carbon into the system. So we obtain now this three-dimensional surface in which we start with an age distribution at equilibrium in the year 1765. We add fossil fuels to the atmosphere according to the RCP 8.5, and we see that we add a huge amount of young carbon, and therefore most of the new carbon is young, and then that carbon starts aging, and therefore it becomes older later on. Basically what this graph is telling us is that if we add a lot of fossil fuels to the atmosphere, then we add a lot of new carbon, but this carbon actually over time will stay for longer time. And on average, the mean and the median age of carbon in the atmosphere will become much older. We can also use this same type of simulations to look at the transit time as it changes over time. I'm not going to show you that in this example, but the point is that with this set of formulas, we can look at how age distributions change over time as we have a system that is in equilibrium. So, to summarize, ages and transit times are powerful metrics to study system-level processes in biogeochemical processes. Ages and transit times are different for complex systems, and their distributions reveal important properties for particular ecosystems. And we can use this theory to ask very specific questions about ages and transit times that integrate many different processes in ecosystems. The calculation of this system-level diagnostics is facilitated by dynamical system representation of models. So therefore, it is very important that we can express models in this matrix form so we can actually do this type of computations. So that's all, and thank you for your attention.